Although, you know, we're pretty casual, so if you feel the need to get snacks during the program, that's encouraged. So hello everyone, my name is Ariel Firebaugh. I'm the Director of Scientific Engagement here at Blandy. Yeah, feel free to move chairs however you like. We're Blandy casual. I'm so excited to welcome you all here for our third program in our Centennial Series. This is a great day. We're here to celebrate a birthday. Blandy turns 100 this year. And so we're cooking up all kinds of centennial celebrations for the next couple of years to celebrate this monumentous occasion. Today, we're here especially celebrating the communities that make Blandy and the State Arboretum such a vibrant place. So we'll be learning from members of our research and education communities here at Blandy. More on that in a moment. First, I'm going to share just a little bit about how today's program will work. So we are live streaming this program to Blandy's YouTube channel. The camera is right over there, hiding inside that box. Hello to the people who are tuning in on YouTube now or in the future. So what this means is that you all can rewatch the program later on if you'd like, or perhaps share the link with friends. If you're here in the library, of course, I hope you found the reception in the back, some little nibbles. Feel free to partake at any time. Restrooms are available outside, so you can go out of the library, walk into the courtyard, weave your way through all of the various photo shoots that are happening out there, and you'll see the restrooms to the left of the archway. So our Centennial program series has been going on all spring. We're going to take a break for the summer, and our next program in the series will be in September. It's called Rare, the Quest for the Uncommon, so all about things that are rare in biology, rare mutations, rare species. I'm looking forward to our September program, so we hope that we'll see you in here again in September. Now for today's program. So I'm so excited for our lineup. Our speakers today include current graduate students, a Bloomberg monitor, Casey Leclerc, who's a member of the Northern Shenandoah Audubon Society and the Virginia Master Naturalist. If you are a friend of Blandy, you've probably seen her around here quite a bit, riding around on the gators, checking bluebird nest boxes. And I know that many of you here in the audience today probably work with Casey on that project. And we'll also be hearing from Blandy's pre-K through 12 director, Harrison Jackson, about the school programs that take place here at Blandy. So at the end of the program, we welcome you to stay for an activity. Might be inside, might be outside. We'll figure that out when the time comes. But an activity, sort of an opportunity for us to learn together, be a community together, and maybe pretend like we are school children here participating in one of Blandy's field investigations. Okay. So I have a few slides just to sort of introduce the program, and then I'm going to kick it over to our stars, our first group of speakers. So today's program is all about community. This is a picture of a single bacteria cell. Now this bacteria is called Vibrio fischeri. I think it's just had a name change. Maybe it was in your biology textbook as Vibrio fischeri. So teeny tiny bacteria, invisible to the naked eye, but when these bacteria come together in high densities, when they achieve a quorum, something really remarkable happens. These bacteria don't have eyeballs like humans, they can't see each other, they can't talk to one another with words, but they can still sense how many bacterial cells are around them. And they do that with this interesting signaling strategy that they have. So here's a representation of one of our Vibrio bacteria. The bacteria divide, and as they divide, they create these signal molecules. So those are the little red circles that you see there. So they continue to divide, continue to generate these signal molecules. When the signal molecule in whatever container the bacteria are in goes above a certain threshold, the bacteria are sensing the concentration of that signal molecule in the environment. And at some point, the bacteria infer that they're not alone. They're part of a big group. And then all of a sudden, something really magical happens. In unison, all of the bacteria change their behavior. When they hit this critical mass of about 10 to the 12th cells per milliliter, they start to glow. It's pretty amazing. So here they are glowing in this flask here. Now there's actually more to the story. It gets even cooler. There's a really cute squid in the mix as well. So that's something for you to go home and Google. But my point here is that there's really power in coming together. Something special happens when we reach a critical mass. And you can see lots of examples of this in the natural world. From these bacteria that glow when they get together, to slime molds sort of slithering their way through the forest floor in search of food, to bumblebees living together in colonies that will actually fan the brood cells to keep the nest cool on a hot day, perhaps like we'll have coming up here in a couple days. So lots of examples of the power of community in the natural world, but we also see lots of examples of the power of community and scientific discovery here at Blandy. So I'll just flip through some of those real quick. Starting at the beginning with Dr. Orland White, first director of Blandy. He had a network of collaborators across the country and probably across the world. They were all trading tips and tricks about how to breed bigger, better tasting pawpaws. Here's another story. This one was new to me. So the little girl that's here on the left, her name is June Lee. Her family immigrated to the United States from Taiwan in 1950. June's father was a research fellow here at Blandy. So you can see that the family in the photo here on the right, surrounded by other members of the Blandy research community. Now, when June arrived here at Blandy, she didn't speak any English. And we have diary records from some of the early students who talk about how they were so happy to help June learn English, and especially learn the words for many of the animals that June might have encountered here at Blandy. So deer, cats, cows, learning all about the natural world and making discoveries. Another fun fact, June's father would go on after his time at Blandy to become the director of the Morris Arboretum in Philadelphia. So now we're going to fast forward to the 1990s. 16 environmental scientists from around the country, including Blandy director Michael Bowers, convened at Blandy for a 4-day workshop. They met just across the hall here in the dining room. And as part of that workshop, they dreamed up a national project that would become the National Science Foundation's National Ecological Observation Network, NEON. If you have tramped all around Blandy, you may have seen the NEON Tower back in the back, but this is a hugely important national network. And the idea was cooked up just down the hall in the dining room. By the way, so this is a scan from an Arborvitae article from the 90s. If anybody has the original to this picture, please let me know. I'm looking for it. I'm desperate. John Porter, I'm looking at you. Probably in your office. Just a couple more examples. Of course, our graduate students work in community, often working with help from some of our wonderful nonprofit organizations in the community, like the Master Naturalists. Kate LaCroix worked with the Virginia Master Naturalists and others to map the distribution of mason bees in the state. Some pretty exciting discoveries there. And finally, young people. So this is such a cool collaboration. Have you all seen the purple martin colonies kind of back behind where Lake Arnold usually is? So, a couple of summers ago, we had a group of young naturalists in our summer camp program. They wanted to study purple martins. And because we had this wonderful purple martin colony, they were able to go and very strategically position their research base under a tree where they could get some shade. And they wanted to know at what times of day are purple martins most active. So this great synergy of community group creating habitat for birds that then feeds into our summer camp program, encouraging these young naturalists to be curious and make discoveries out there. So that was just a lot of examples of some of the communities of discovery that stuck out to me when I was thinking about this program. And I'm so glad that you all are here to celebrate communities of discovery at Blandy today. So I think I should stop talking. One thing that is special about today in particular is that this is move-in day for the students who will be participating in our summer research program. So I thought it would be appropriate to hear from some of the students who are part of our research community. To me, the students are really the beating heart of the summer research community here at Blandy. So I wanted to make sure that they got the stage first. So with that, I'll invite up our three graduate students who are here in the front, Ethan, Willow, and Mia. We've got 2 chairs. So we can work that out. I think I'll ask you to just go ahead and share with the group who you are and who you work with. Just a couple of thoughts about your research system. So what are you studying here? What does a typical day look like for you? And then if you have any words of wisdom about what it means to you to be part of the community here, or even just a story that sticks out to you, you can share that with the group. And I guess we'll start with Ethan, and then we'll do Willow and Mia.
Sweet. So hi, I'm Ethan Sketches. I'm a now I guess second year PhD student at the University of Virginia, and I work with Dr. Megan Blumstein. And broadly, we look at forest genetics. My research is really honed in on the idea of assisted migration and how plants are responding to climate change. And so we see them kind of shifting their ranges farther north. I work with black gum trees specifically, and so a lot of that right now is traveling up and down the range of black gum, which is from like just east of the Mississippi over in Arkansas to up to New York and kind of stretching up into Vermont areas. And I'm slingshotting leaves out of trees to do genetics on them. And so it's a good time. We have like, I would say like an 8-foot tall slingshot that we use. And yeah, it's nice to get to be outside. My research is still kind of projects growing, developing, but that's kind of what I work on while I'm here. I think in terms of being here, it's so nice to be able to get out of Charlottesville for a little bit. I love Charlottesville, but it's nice to get to come to a place that feels a little bit, I don't know, we kind of use like summer camp rituals in a sense, but you're just surrounded by like-minded individuals. Our program at UVA is great because it's really interdisciplinary, but then these summers allow us to kind of like really build community within the people that study the same sorts of things, whether those be pollinators or plants or that sort of thing, and kind of grow that community outside of the Charlottesville area as well, and kind of have another place to call home within Virginia if we're not from here, which I'm not, and so it's lovely to kind of have Charlottesville as a home base and then also up here in Boise.
Well, I'm Willow Lebecki. I am going into my 4th year of my PhD, which is crazy. I work with Ty Rolston. My research is focused on nesting biology of bees. So I work out at the bee wall, so all the chimney bees that are going crazy right now. You might have seen like the little mesh cages that are over them. That's me putting them out there. Yeah, I'm interested in generally nesting biology, nesting costs, the materials that we use, and and also like what it means to live in a group. So kind of thinking about the bees community. Yeah, and Blandy is a very special place to me. I grew up in Winchester, so I've been coming to Blandy since I was like in preschool. So I came here as a little kid on school trips, and then I actually also became a master naturalist in this very room when I was 16, 10 years ago, and then started working with Ty in high school. And so Blandy, I went, then did my undergrad in Wisconsin, came back to Blandy because of the community that's here. And yeah, as a graduate student, it's been awesome to be around other graduate students. The community here in the summer it's like alive and full of ideas, and everyone is very excited to be, when we are in Charlottesville, it's a lot of like, we're at our computer and thinking about the things we're gonna do, and then we're here and it's like, oh my gosh, it's so busy and exciting. So yeah, it's wonderful to return to a place that has always felt like a really welcoming and loving community.
I'm Mia. I work with Ty Rolston. I am now going to my second year of my master's, and I study the native butternut tree, which is part of the walnut family. And so the native butternut tree ranges up and down the East Coast and has really had like really deep cultural connections to people for a long time. And unfortunately, this non-native fungal pathogen called, well, there's a scientific name, but I call it OCJ. It causes butternut canker disease, and so it's completely decimated the populations across most of its range. And for whatever reason, a lot of those monitoring efforts have not included Virginia. So I'm working this summer to try to find as many remaining butternuts as I can in the wild here. And so I'm working with incredible citizen scientists across the state, Virginia Master Naturalists, Virginia Working Landscapes, and so many others, including Casey. And so I am looking to find trees, get a sense of the disease severity there, measure them, collect leaves for genetic sampling, in hopes that, not during my time, but eventually that Blandy can have a tree breeding program that is actually propagating trees that are resistant to the disease. So that's what I work on. I actually came to Blandy a couple years ago as an undergrad researcher working for Ty, studying native bees, and then have been here as a technician and now here as a grad student. And getting to be here at so many different levels, I think Blandy is one of those special places that keeps people keep coming back across career stages and across, even if your research questions change. And so when I was here as an undergrad, I was able to work with high school students, and now as a grad student working with undergrads, and just continue to build that network. And when I go off and go to conferences, I somehow continue to meet people who are like, oh yeah, I worked at this random farm in Virginia, you've probably never heard of it. And I was like, Blandy? And they're like, yes. And so this network is just so expansive. So even if I don't I'm not here at the same time as people, getting to go out into the world and continue to meet people and just continue to build that community has been really special and really, really awesome.
Well, thank you, students. I know, I really appreciate that the summer is crunch time for field research. So I so appreciate you all for taking a couple minutes to share about your experiences here with the group. So thanks so much for being here. I'm going to release you to your next adventurers. And with that, we're going to call up our second speaker, Casey Licklider. I'm going to read your bio. Do you want to stand here or do you want to sit down? Do I want to what? Do you want to stand here or sit down while I read your bio? These are words that Casey wrote, by the way, so I don't think I'm embarrassing. So excited to welcome Casey Licklider to the stage. This is what Casey says you need to know about. While raising 2 daughters and working full-time, Casey's love of nature inspired her to earn an associate's degree in National Resource Management from Laurel Ridge Community College in 2005. She studied with the Institute for Bird Populations and gained experience at various bird banding stations to become a master bander permit holder. She established and operated a mist netting station with the Monitoring Avian Productivity and Survivorship Program for 5 years, located at the Burl Van Lennep Foundation. Did I say that right? Casey currently serves as community coordinator for Virginia Bluebird Society for Clark, Frederick, and Shenandoah counties. She has held office of treasurer for Shenandoah Audubon since 2012 and serves as their conservation committee chairperson. Casey is the Shenandoah Audubon / Blandy Purple Martin Colony landlord, as well as trail manager for Blandy Squirrel Farm, Bell Grove Plantation, and Seven Bend State Park Bluebird Trails. Casey is a pencil artist, has 4 grandchildren, and shares her home with her dog, Ren. What a lady. Casey, thanks so much for taking the time to speak with us today about your work with the Bluebird Monitoring Trail. And I'm going to call the presentation.
Hi everybody, thanks for coming today. So when somebody wants to know about bluebirds, I get really excited. So I'm on a time limit, so we're going to go really fast. If you have any questions, feel free to approach me afterwards. I think Errol's going to give us time for a few questions. So here we go. We have Blue Wildlife on Hawaii, the Northern Shenandoah Valley Audubon Society Bluebird Trail here at Blandy. We all know the species started declining in the 1900s due to loss of habitat, competition from non-native bird species, outdoor cats, and some of the things that we did. So when people started noticing the decline, they started putting up nest box. So if you go across the United States, transcontinental, you've got nest box systems. Bring it down to the state level, you've got the Virginia Bluebird Society. Bring it down further than that, you've got our chapter of the Audubon Society. Bring it right home to Blandy. Right here we've got 132 boxes. The trail was started by Charlie Clevenger back in the 1970s, 80s. He started putting up nest boxes, and he ran into Sam Patton around 1994, and they joined forces. And that's Sam Patton and Charlie Clevenger, and there's me following Charlie around. In 2004, this is Greg Baruffi, and I visited Charlie, or Sam, and Sam asked me if I would take care of his bluebirds. I said yes. I didn't say for how long. It's been 23 years. And I've got a lot of help, an excellent team. Everybody who's on the Bluebird Trail family, raise your hand. Okay. And how about the Purple Martin Colony? Okay. So Greg and I co-managed the trail until 2008. He passed away in an automobile accident. But before that, we established our trail objectives. We started putting our brains together, and we decided we wanted to provide habitat, educate people, and do what we could for research. So the first thing we did was we took off and we drove to Cornell Lab one day. And we met with one of the research assistants up there, and we came back home and we developed our protocol map. Okay. So then we thought, okay, we've got to take what's out here in the field, we've got to totally renovate everything, had boxes on the ground, we put predator guards, the stovepipe here, and we changed the wooden posts to metal. We designed a map, it looks like a bunny on a stick, and each year we evaluated how many boxes we could fit into Blandy, like a little incubator. We went from 100 boxes in 2004 to our current 132 boxes. We designed a nesting chart for bluebirds so that people could actually visually see the days of the month, how many days they lay their eggs, how many days they incubate. And then if they figured out the first egg date, they could go to the critical nesting period chart and figure out when you can look and when you can't, for safety for the birds. We designed our own worksheets for the monitor when they go out in the field, set up our trail books, and we started our workshops. Lo and behold, front seat, Sam and Charlie. So the species we focus on, the native birds, tree swallow, bluebird, Carolina wren, house wren, and Carolina chickadee. People learn to identify the species by the nests and by the eggs. This is what they get to see about the bluebirds. They see the eggs hatch, they see the chicks grow, and the birds fledge. So our contributions to research. For the Virginia Bluebird Society, we did color banding and applied the USGS band for survivorship and nest site fidelity. For Cornell Lab of Ornithology, we did an incubation rhythm study where we put little dime-size temperature gauges in the nest cup and in the box, and it would produce a graph. When you plug it into the computer, it would produce a graph that shows the ambient temperature in the nest box, and then when the bird leaves the nest, the temperature drops, and it showed how many times the nesting bird left to go out to feed, and then the temperature would fall. It showed how many times it came back in to sell the eggs. So Cornell took all that data. We had to submit everything in handwritten form with these little cards. And they did some studies on it. And we were working with Dr. Kieran Hooper and Tina Phillips. They produced some articles. And then Dr. Cooper came to Blandy. And she gave her talk on citizen science work with Cornell Lab, which was really cool. And then a reporter came from Science Magazine and interviewed Greg and I, and he was doing the report on citizen sciences. So we were in the same issue of Science that the Ivory woodpecker was in. That was in 2005. So that created some excitement. And we're always on guard against the European house sparrows. They're aggressive. And we're tracking the number of house sparrow eggs that we take out of the nest each year. Some years we don't have any, and other years it's like over the top. And the doctor from Cornell moved to North Carolina. And so when she gets to North Carolina and reads up natural history, she says, calls us up, do you want to be involved in the sparrow flock project? So I said, yes. So we gathered up all of our sparrow eggs, wrapped them in little pieces of paper towel, put them in a little plastic egg, and put them in an egg carton, and shipped them down to her, where they did studies to see where heavy metals detected, and they were looking at the eggshell thickness and the coloration of it. And the last I talked to her last year, she didn't have her final paper written yet, but I'm on the lookout for that. And just simple things like finding horsetail hair in a nest. Well, it can be really dangerous for the chicks. And we discovered this, and I wrote an article on it, and it went into the North American Bluebird Society Journal. And then somebody had questions about this same thing out in Oregon, and they were Googling and found the article, and then they produced it in their newsletter. So all of this community really goes across the country. And then there's a gentleman back here, raise your hand, Dave. He and I discovered a mystery bird on the trail one summer, 2021. It had actually chewed around the hole, the entry hole, making it larger, and created sawdust, and then laid 2 eggs.
Wow.
And Dave and I at separate times would hide out in the bushes out front to see if we could figure out what bird this was. We finally figured it out. It was a red-headed woodpecker.
Oh.
So, and then I wrote a paper about that, and the Bluebird Journal picked that up again. And I don't know about Dave, but that was like Christmas for me. trying to see what this bird was. So we've been tracking the fledglings per year since 2004. And they kind of fluctuate a little bit. Our worst year was in 2010 when we had 97 fledglings. And if you remember, that was the year that we had the great big snow. And the ground cover was like forever. So that affected. And then 2024, they have 438. That's a lot of eggs. Also track the tree swallows, of course. We track each species that we cater to. The lowest was 149 in 2007, and the highest was 570 fledged in 2019. So the number of fledglings since 2004, 14,370 that we track as fledged. And all of this data each year has been entered into Cornell Lab of Ornithology. These are the people that have become trail technicians. It is teamwork and it's a family to me. And I'm so happy to work with all of it. And there you go, you never know what you're going to see out on the trail, who or what. So if anybody's interested in ever participating in this project, we'd love to have you. Locally, these trails have developed as offsprings from Blandy, whether it's someone, a private resident, got a couple people here that have created their own trails at home, and state parks. So let's see here. That's it.
Thanks so much, Casey. And we do have time for some questions before we pass the baton over to Harrison. I'll get us started, Casey. Besides the mystery bird, what was one thing that you've encountered during your time with the Bluebird Trail that surprised you?
One thing that surprised me? Well, I don't know about surprises, but I think maybe the best, I guess you might call it a surprise or a gift, would be the way everybody works together. It's really a lot of teamwork.
What a great transition for our program about communities. Couldn't have played that better myself. Questions from the audience?
If we wanted to get involved, how could we do that?
Contact me.
Okay.
And you can, Ariel has my information.
Okay.
Yeah, I was curious, what is the recommended acreage per nest box?
I don't know about the acreage,
Yeah.
but let's say it should be like 300 feet apart or 100 yards. But that's not always necessarily the rule either, because Blandy here has a lot more than that.
Right.
At my own house, I have a little over 3 acres and maybe most of it's wooded, so most of my boxes are out in the open. And I have like 5 boxes. So, and I think when you have the boxes closer to each other, different species can nest without competing for the food. Like you can have a tree swallow in one, and a bluebird in one, a chickadee in one.
Right, and they'll all play nicely.
Not always necessarily,
Okay.
no, but you know, that's nature.
Thank you.
Why is horsehair tail dangerous for fledglings? Because on our farm, we found nests all
I'm
the
sorry, I
time.
can't hear you.
Why is horsetail hair
Oh.
dangerous for fledglings? Because there wasn't much you could do about that on our horse farm.
Okay, so the horsetail hair in a nest cup is kind of like plastic string. Because it's like, I don't know what it's made out of, I guess some kind of keratin. And it's really, really strong. So when it gets all wound up in that cup and then the chicks get in there, they get turned and they get their feet caught and it gets wrapped around their wings. And in this instance, we had 3 chicks in the box. And when we went to monitor the box, one of them was actually upside down wrapped up. And so we took them out, we unwrapped the chick. And then I unstrung all of the horsetail hair and put the chicks back, and 2 of the chicks survived. But the one that was all wrapped up, he didn't make it.
In your monitoring, can you get to the nest before the eggs are laid and remove horsetail hair?
Anytime you find it, yeah, if you can do it without tearing up the nest.
Other questions for Casey? Well, if you think of one, we'll have lots of opportunities to chat with Casey and Harrison after the program. Casey, thanks so much again for sharing your experience. I know I'm looking forward to looking up that science article and getting your autograph. So I think we'll switch gears and we'll hand the baton over to our final speaker for this program, Harrison Jackson. So Harrison has always had a passion for the environment, spending his childhood summers on the Chesapeake Bay and in forests and fields in Maryland and Delaware. His love of nature and natural curiosity led him to attend Salisbury University for his bachelor's in marine biology and later Clemson University for his master's in wildlife and fisheries biology. His enthusiasm for the great outdoors and dedication to ensuring all students have opportunities to learn and explore outside led to a career in environmental education, where he's worked for nonprofits leading K-12 environmental education efforts for over 15 years. His work has taken him across Maryland, Delaware, North and South Carolina, and now Virginia, where he's the director of K-12 education at UVA's very own Blandy Experimental Farm and the State Arboretum of Virginia. Harrison, the floor is yours. Thanks so much for taking the time to speak to us.
All right. Thanks, y'all. I appreciate it. I apologize if you see me moving around a good bit. I'm used to teaching and doing things outdoors, so sometimes staying in one place inside can be a little challenging for me. So I appreciate y'all coming out today. Thank you for the introduction, Ariel. We're gonna go ahead and continue to talk about basically how we strive to really grow lifelong learners here at Blandy and continue to have them ask questions, dive into the scientific process, and really connect with nature in the way that students are really supposed to, but still igniting that natural curiosity. All right, so Blandy, we all know, is a really special place, right? Bequeathed by Graham Blandy in the 1920s under the auspice that we would continue to do research and education on agriculture. Well, we've changed a little bit in that 100 years since Graham Blandy bequeathed it in the 20s, but we still continue to do a lot of research and education on now things beyond agriculture. So Blandy's mission is to increase the understanding of the natural environment through that research and education. And we do it in really 3 main ways, right? 3 main pillars. We have our university research faculty and staff, as well as our undergraduate and graduate students who y'all just met earlier today. They're out here in the summertime. Our research faculty are out here year-round, and they're really doing a lot of different ecological research, focusing on different plants and insects and birds. We also have the State Arboretum of Virginia, so those central 170-ish acres. That's really important. It has a really amazing living museum of specimens here that we can utilize to showcase not just these really amazing plants from around the world, but the fantastic plants that we have here in Virginia. And then finally, we have our outreach and education programs. So one half of that is myself. I work with the kind of formal K-12 programming, so all the schools, which we'll talk about here in just a second. But Ariel also is one half of that as well, and luckily she takes on the parts that are much more challenging with adult public programming, the Ginkgo event, which is lots of fun, and lots of different other opportunities to bring in and kind of create this community of learning here at Blandy beyond the traditional research and kind of K-12 education. All right, so education programs, we've been doing it for a while here at Blandy. This is Mary O'Lean, who is going to be our president, right, of the FOSA board. So she was our education director way back in the day, in the 1980s when we first started. We've been doing this for about 40 years. Every year we see about 5,000 K through 12 students. They bring about 1,000 chaperones. That number can increase a little bit depending on the grants that we get and some of that extra capacity. So sometimes we get up to around 6,500 students, but we usually keep around 5,000 or so. We have about 350 teachers who come out either as part of those field investigations and field trips or as doing professional learning. And like I said, we've been doing this for about 40 years, and since 2006, we've seen about 93,000 students. So a good number of students have come through Blandy education programming. And we do that with a lot of different partners. While we certainly like to focus up here in our corner, we work across the state. We are the State Arboretum as well. So the primary districts that we work with are some of our closest: Warren, Winchester, Clark, Frederick, Loudon, Fauquier, right? We see and work with a lot of those students, right? Clark County students, we see them quite a bit. We'll oftentimes see them in 3rd, 4th, 6th, 8th, 9th, sometimes even more than that. So we'll be able to see them a good number of times. But we do a lot of work with students, and really especially teachers across the state. We really like to make sure that teachers have the resources available to be able to take their students outside and get them engaged, whether or not they're here at Blandy. Although, of course, we would like them to be here as much as possible. And then one of the big things that we really did to incorporate this work was we work with and make sure that all of our programming is aligned to the Virginia State Standards of Learning, which basically means that everything that they're going to learn out here, all the vocabulary, all the concepts and abilities and skills that they need to have, are going to be mirrored in the classroom. So that way, everything that we do isn't just a one-off. They're not coming out here to learn about pine trees. They're learning here to learn about ecology, and that fits into the unit that they're already learning about, and it's going to be all scaffolded in, and then when they see that at the end of grade test, that's always stressful, right? They're going to remember everything that they learned here at Blandy. We hope, at least. That's the plan. So what I really want to talk about today is our main framework for teaching that we call inquiry-based learning. So what this is, and I'll talk about more in just a second, is the framework that we use to kind of make all of our lessons, our curriculum, and to teach. And so I really like this photo. It's kind of hard to see what it says, but this is some of our high school students from Skyline. And the question they were working on was, what physical similarities are there between some of our insects found on sneeze weed? So they were able to do a study and look at this methodology and share it out with their classmates to talk about some of the things that they found out in our fields. All right, get ready for the text-heavy slides here. All right, I'm going to talk to y'all a little bit about kind of the basic framework and then dive into how we develop some of this lesson planning, right? So what is inquiry-based learning? Well, this is going to get a little metacognitive here, but it's really rooted in constructivism learning theory, which basically means that as humans, right, the things that we learn, everything that our knowledge and our meaning is taken away from the experiences that we have, right? So it's much easier for us to understand the things that have happened to us versus something that somebody else talks about that's happened to them. Pretty simple definition. Inquiry-based learning, right, this is from Lee et al. From 2004. It's a little bit of an older definition, but I really like it. Basically, it's an array of classroom practices that promote our student learning through guided and increasingly independent investigations. Oftentimes, which there's no answer, but there's a lot of complex questions and different procedures. And so, effectively what this means is this kind of classic Socratic didactic lecturing method that I'm doing right now, where I am imparting all of my information to each and every single one of you and hoping that all of that sticks in your brain, isn't necessarily the best way of teaching, right? We call that the stage and the, but what we've noticed is to the kind of educator leader and kind of move them into a facilitator of the learning experience, right? So more of a guide on the side. You're really there to help facilitate and help those students learn, right? So I might be asking a lot of questions, right? But I am not going to be telling the students all this background content. It's up to them to explore some of that. They're going to be really exploring this data analysis, and they're going to be drawing the conclusions and sharing that out and communicating with the rest of the group. So why do we do it? Because it's really fantastic and it's fun. But beyond that, right, it's really a fantastic way to take our students and turn them into contributors of knowledge, right? It's not just them taking in all this information. It's not just them scrolling through TikTok or Instagram, right? Now they're able to actually go out there and say, hey, I contributed to this study. I was able to go out there and figure out this, right? It's just awesome. They take more active role, they're more engaged. It helps a ton for them to build up their critical thinking and problem solving skills. It really builds up resiliency. One of the main things that I've heard a lot of teachers dealing with, and I think we might also see some students, younger students dealing with, right, is that they come across a problem, they get frustrated, and they don't know how to take that step back, look at the larger problem objectively, and then figure out a way around that, right? So how do they build up that resilience? It's a lot easier to do that when you're younger than when you're 30-something, right? It also helps them to build up those social and emotional or soft kind of skills, right? So communication skills, leadership skills, project management, all that kind of things, right? It's gonna boost their confidence, get them more engaged in STEM, especially girls, right? We know girls, especially after grade 6, their interest in STEM really tends to drop off. So this IBL really helps to keep them engaged. And then it also allows them to have more intellectual risk taking. All right, so I love this little picture of one of our littlest students here drawing something, not quite sure, probably a flower. But what we're gonna do is I wanna go ahead and show you how we kind of go through our levels of inquiry, build out our IBL lesson planning, and then develop out some of our question strategy. All right, so our levels of inquiry. When we think about inquiry-based learning, we have to first think about how do we actually want to guide this experience, right? I can't take that little kid right here and plop him out into our research field or at the B wall like with Willow and say, great, let's do that dissertation, right? That's not fair for this student. They don't have those skills built up yet. So how do we do that? Well, we do it gradually, right? So hopefully, or maybe some of us all know how to swim, right? And hopefully maybe some of us can remember how we learned to swim, right? So in this metaphor, I like to think about, right, is how we learn to swim. And so it's the same thing for our levels of inquiry. It's a spectrum. So on our left-hand side here is our structured inquiry. This is, the teacher has the most say in this. They're really helping our students learn, but the students aren't gonna have a lot of free kind of license to do whatever they want, right? So think about a metaphor if you're learning to swim. They're in the pool, they have their hands up on the side of the pool and they're kicking their legs, right? But they don't have a lot of license to go out and do anything else, right? So whatever our inquiry is, whatever investigation we're gonna be doing, we're gonna have it really on guardrails. The next one is gonna be our controlled inquiry, right? So this is gonna be an investigation that's a little bit more open. Our students have a little bit more of some leeway and some choice on what they wanna be able to do. So thinking about our metaphor here of learning how to swim, now those kids are off of the wall, we give them some kickboards, and some of those kids are gonna do great. Some of those kids are going to start to sink a little bit. We have to monitor those kids a little bit more. But, right, we're allowing them to have a little bit more choice and a little bit more independence. Finally, we start to get over into our other sides over here where we have a little bit more openness and some more student voice. So our guided inquiry. By this point, when we're thinking about a guided inquiry investigation, I, as the teacher, might just ask a question, but I am not going to give a lot of other guardrails to my students, right? So no, I'm no longer giving them the procedures, the methodology. I might be asking them something along the lines of, if we want to try to investigate what is the healthiest watershed here in Virginia, right? Very broad. How would they investigate it? What questions are they going to start looking at? What data do they need to draw the conclusions necessary to make that answer? So in our analogy here, now they're going to be swimming by themselves, right? They're going to be all going ahead and swimming in their lanes. They might be, some of them might be doing breaststroke or front stroke or backstroke, doesn't really matter. They have a little bit of choice, right? They can choose. And finally, we get to free inquiry. I am no longer, as an educator, really doing anything there, right? So we see this a lot with our researchers here at Blandy, our faculty members, our graduate students, our undergraduate students. They're out there doing free inquiry. They can choose whatever they want to investigate. For us here at Blandy, it's a little bit more challenging to do that with our K-12 programming, right? So when we think about our inquiry-based learning for our K-12 programming, what we really want to do is focus a lot on these sections right over here. We do that for a number of reasons. Right? A lot of times we work with students from across the learning spectrum, and so they might not have the necessary background skills to be able to come in here and do an open inquiry. But part of it also is a constraint in time. So we love to be able to work with some of our high school groups, and sometimes we have the capacity to have them come out here and do a year-long curriculum or a semester-long curriculum. And there are other times when we have students out here and we only see them for 2.5 hours. So it's really challenging to do a 2-week investigation when we only see them for 2.5 hours out here, right? So sometimes it's just a little bit easier to keep some of those guardrails on to say, hey, we're gonna go ahead, we already have the procedure and the tools that we're gonna use. We're just gonna let y'all choose maybe where we go and collect some of those samples. Right? So just a couple examples of what that might look like here. This is a picture of some of our 3rd grade students. They are in our soil program. We love having them out here to look at all these different types of soils that are native to Virginia, some different soil profiles that they might experience. Right? The experiment that they're looking at is a soil porosity experiment. So they're trying to figure out exactly how quickly some of this water is going to go ahead and percolate through some of that soil. It's class-wide, but it's very, very, very structured. We have all these rails on here. We have the methodology on the whiteboard. We have our chaperone watching over everything. We have all the amounts that they need written down, right? So this is exactly what you would see in a classroom typically. They don't have a lot of voice. They don't get to say, hey, Mr. Harrison, do you mind if we try this other different type of soil, right? Because we don't have that with us. We don't have the capacity due to time, right? So we already know the answer. The answer is pretty much prescribed. But right, those students are still going to have that chance to really investigate and get to that conclusion themselves, which is what we want them to build out to. Right? And they can work in groups and they get to have some of those discussions. And we really want them to encourage them to have those discussions in groups. So we typically tend to have a lot of our work done in small groups or in partners. So our next level, our controlled inquiry, we're trying to take away some of those guardrails a little bit for some of our students. Now it's a field observation study for some of our high school students in our algebra functions and data analysis classes. And so what they do is they'll come out here and we'll be looking along a 50-meter transect that goes out into our fields. And they'll be measuring soil moisture, pH, and then also plant biodiversity. So while they still have to measure those things, right? We're working along a 50-meter transect. They get to figure out where they want to do that along that transect. Where do they want to collect some of these samples? Which samples do they want to collect? Who's doing some of that work? Right? So they get a little bit more of this voice and this choice in where they want to do some of this. But we still know at the end of the day we're going to have 5 sample sites that they're going to be taken from. We're still going to, they're going to have 10 plant samples probably, right? So there's a lot of the same information that's already prescribed. And then finally, for our most open inquiry, are our guided inquiries. These are ones where we're taking off pretty much mostly guide rails. The only thing that we're asking them is usually a driving question. So some of our high school groups, like that Skyline group that came out to measure some of that insect diversity in our sneeze weeds meadows, as well as our MeWe investigations. So this is a meaningful watershed education experience. And so these are a year-long curriculum that we'll see students out for where they investigate something like a watershed. And so it's a bigger project where we get to have them a driving question. Specifically, this was Clark County. This was 6th grade. We were asking them how healthy is the Shenandoah. And they were able to then, at the end of that year, take everything that they've learned, right? They would go out to look at some of our water quality samples. They learned about land use and some of the different ways that land use impacts water quality. And they were able to go out and do a student-led action project. So they decided what they wanted to do, which is help with this stream restoration, right? The teacher was there mostly to just help out, order things, make sure that they had the plants and that students weren't hitting each other with shovels, right? All the things that teachers are supposed to do. Well, all the students worked together in their class to say, hey, I want to take on ordering the plants. We need to figure out which plants work best closest to the water, which plants work closest, work better further to the water, right? Where are some of our sunnier spots? So that's really up to the students. Will some of those plants and some of those gardens fail? Absolutely, right? But it's a learning experience for them to understand some of that, and the adults are going to help with some of that feedback to say, here's where you might want to think about using a different species. Right? It's still ultimately up to the student's choice. So it allows them to have very, very light guardrails in place so that they can still fail, learn from that failure, and then grow. right? Which is ultimately what we want our students to do. We want them to ask questions and understand that it's okay if they make mistakes. So now that our students, we've just figured out what level of inquiry we want with our students. We want to have it really open, we want to have some more guardrails, really decided for our lessons. Right, I'm going to go ahead and talk a little bit about our 5E model. So this is an instructional framework that we use to develop majority of our lessons. And allows us to kind of sequence what we do for some of our programs. Right? What this is, it allows us to kind of basically show, how we start using an engage piece, and then kind of go out and go from there to figure out how we want the students to investigate and explore the rest of that phenomenon. So the engage piece starts out, the 5E model starts out with the engage phase. That piece basically starts out with a question, a phenomenon, or an activity that gets the students interested. That can be something that just makes them curious, that can be something that is a bigger kind of question. From there, then the students are going to go ahead and investigate that phenomenon or that driving question. They're going to go out and explore and actually collect some of that data. So they're going to use some of the different tools, techniques, and procedures to collect the data that they were looking for, and then explain, moving into that explain phase, to go ahead and use that data to explain, answer the question that they were originally looking for. So, a lot of the times, these kind of lower level, inquiry level programs, where we have our students doing really prescribed activities or investigations, they don't really tend to have a lot of the other phases of our 5E model. So they'll start out, they'll ask the students a question, they'll go out and they'll collect their data. The students will come back and they'll use that data to explain the original question. And that's great, that's fine, right? But what we really want to see is some of these other sections being used are elaborate and evaluate, and I'll say that reason in just a second. So an example of this in action. Okay? Here at Blandy, we do a lot of different programming around the watershed. Right? So just an example, if we were creating a new lesson and we're thinking about how we want to sequence that so that our students understand and can build out their investigation properly, we'll start with that engage phase. So we'll ask them, right, what is or is the Shenandoah River healthy enough for us to swim in? Right? So what we want to do is engage a lot of different background content with them. So now they're thinking about, okay, what do I know about the Shenandoah River, right? What's in that river? Where is the river? Another thing they're thinking about is how does my family utilize the river, right? Do I go out fishing in it? Do I swim in it? Do I bird in it? And then finally, they're also kind of creating a connection with what do I need to make sure that this water is safe enough for us to swim in it, right? What does that mean to have water that's safe enough for myself to swim in it or for wildlife to swim in it? So they're thinking about and utilizing all this different background content to get them engaged and excited about learning. The next thing that we do is then we say, okay, what do we need to figure out if this water is safe? We need to go out and look at water chemistry. So we'll take a bunch of samples of the Shenandoah River, and we'll have students come out and look at D8 or DO, pH, turbidity, a lot of different types of water chemistry assets, and so that we can figure out, okay, again, did that answer this question? Maybe, maybe not. So if not, we can go out and we can conduct a macroinvertebrate survey. So we can look at a lot of different macroinvertebrates in our water quality to say, okay, or a water sample to say, okay, if we only see rat-tailed maggots, right, maybe our water quality isn't great. If we're seeing a lot of water pennies, water scorpions, damselfly larvae, things like that, right, maybe it is healthy enough for us to swim in, right? So we're starting to be able to explain what that information actually meant, right? So we can extrapolate and understand a little bit about how the data that we collected goes back to our original question. And I think that's really important. But what really makes this a fantastic part of the scientific learning process is that elaborating. Because what this is a really big learning cycle, right? It's basically the scientific cycle. We have them forming a hypothesis, going out collecting data, right? Using that data to either confirm or deny their hypothesis, and then they're going to take that information that they gathered and apply that to novel and new circumstances, right? So they're going to figure out, okay, if I use those macroinvertebrates as a bioindicator, are there other animals or species I can use as a bioindicator, right? Like things like other bird species like bluebirds, right? Can I figure out if that water quality is negative, really heavily impacted by some land use in our area? Is that new data center that's going in with the Avtech Superfund site going to cause a problem for the Shenandoah River and our ability to swim in it? So that is then going to ask more questions that they can go ahead and use in some different data analysis and data exploration tools to investigate. And I think it's a really fantastic way for us to be able to utilize student learning and their own natural curiosity to get themselves engaged in the process. The reason it's called the 5E model and not the 4E model is because it also has that evaluate section in there. Evaluation is always, when people think K through 12, is always thinking about like tests and grades and rubrics and things like that. I don't really like to think about it in that sense as much, because what we're really just trying to see is, does this student understand some of the content knowledge and skills and abilities that we were trying to get them towards, but also are they still investigating? Are they curious, right? So instead of using a summative assessment at the very end of a program, being able to use lots of different questions throughout the program and during the rest of the curriculum and lesson is a really great way to figure out if that student has a lot of misinformation or misconceptions, and it allows them to kind of formulate and kind of rebuff their idea against other individuals, which I think is a really fantastic ability. All right. So once they figured out their level of inquiry and we've gone through our 5E model, the last thing to do is to ask a lot of questions. All right, more fun visual slides. So what this is Bloom's revised taxonomy. This is a hierarchy of cognitive skills, and I don't want to bore y'all too much. Basically, this is how teachers think about student learning and teaching. All right, so all these words you will see constantly in all of the standards across the state. And basically, it kind of shows that you start conceptually here at the bottom. All people, all learners, it doesn't matter who you are, whether you have a PhD or you're a student, just a kindergartner, we all have to just start with basic, simple recall, right? You can't learn about something unless you're able to recall the information first. Can you give me the definition of what the moon is, right? Without that, we can't then go on to the rest of this cognitive hierarchy. And then understanding, right, what is the moon? Where did the moon come from? So these are our base levels of cognition. After that, we start to go up to this middle section, which is where a lot of work happens in K through 12, which is analyzing and applying. Can we take the information that we know and apply it to new information? And then can we also draw some different conclusions, right? So I know that the Earth has a moon. Do other planets have a moon? Yes, fantastic, okay. Now that we understand some of how we're going to be applying it, the students at the very end of their learning, of their cognitive learning cycle, are going to be evaluating and creating with some of that information. That's really hard to do. It's hard work to use your brain to evaluate and create things. And so it takes a while to get up here, right? So students might learn about the moon, right, in kindergarten through 3rd-ish grade, but they're not thinking about how to evaluate the differences between Io and our moon, and critically understanding, right, the difference in substrates between those moons until they're in high school or college, right? So it takes a very long time for them to get up this level of simple recall, just creating those very basic neural networks of understanding, to then going up to being able to apply that information, to evaluating information and creating some of that, and creating new and novel information from that. So I bring all of that up to talk about the way that we ask questions. Kids ask a lot of questions, which I always love, and they ask some absolutely interesting and crazy questions. I've heard some weird ones over the years. Particular ones that always stood out was, do ticks get ticks? Which I think is an interesting one. I also heard most recently, which is why is that rock so small and why is that rock so big? From a kindergartner, right? So thinking a little bit about how do rock sizes change if they're inanimate objects, right? For a kindergartner, it's kind of mind-blowing. I've also had a lot of questions as kind of doing a lot of my grad work in ornithology from students about whether or not birds fart, which I always thought was really interesting. But they're valid questions, right? And so it's something that we kind of sometimes, unfortunately, kind of beat out of students, which is making sure that they just understand. We want them to answer the question, have the knowledge, and be able to answer that on the test, right? Instead of asking more questions. All the time I want students to ask me questions. Even if I don't know the answer, that's really great. We can all figure it out together, right? That's what makes it a community of learning. So how do we utilize and do that? We do it in a bunch of different ways. Sometimes we prepare a bunch of questions. We'll all sit there together in the education room and just think, how do I want to ask a question to a high school student, which can be sometimes a little scary, asking them to figure out and look at soil moisture and pH in relation to this plant biodiversity, or how are we going to think about how that correlates, okay? And sometimes those questions are going to be in different cognitive levels. So we're going to be sometimes in that very, very low level here in the pyramid. Remember, understand, and apply. Asking for some really formative questions, right? So when we have students come to us at the very beginning of the day, if our entire program is about watersheds, the first thing I'm going to ask them is, who can tell me what a watershed is, right? What do we think about when we say the word watershed? If I have a bunch of blank stares and a student says that's where the water is, then I know that we might need to start with the very basics to go over that very conceptual understanding of what a watershed is before we dive into land use and its impact on the watershed. But if we ask that question and they're all sitting there talking about how it's an area of land that has all this water moving towards one specific area, fantastic. We can go ahead and move on to some of our higher level questions, and we can really start to ask some of those questions looking at analyzing and creating and evaluating different ideas. And that has a lot more opportunity for discussion, because it's usually not just one word or yes or no questions anymore, okay? All right, so like I said, we want to have them question everything. And beyond that kind of cognitive hierarchy that we were just talking about, we also utilize a lot of different really narrow, open questions to kind of give them different opportunities to talk and have people have different ways to speak. So when I give them, the students are a little bit quieter opportunities as well. So a narrow question really has only a limited number of acceptable answers, right? So what's the soil, or what's the pH of the soil sample? And then you can also have some really broad questions. So, for example, what stood out most during our nature walk? And that can also be mixed up based off of our cognitive level, or cognitive hierarchy as well. So a narrow, lower level cognitive question might be, what color flowers have the most bees pollinating it? Right, really simple. Student can be able to answer that pretty definitively. A lower level broad question might be, what did you notice most about our meadow? Very broad, very open, allowing students to bring in a lot of different background information. Some people might notice birds more, some might notice plants more. A higher level, narrow question could be rate these different types of bird beaks based on their ability to eat fish. And they should be able to analyze and understand that something like a pelican or an osprey, right, that beak morphology, should be able to eat a fish better than something like a cardinal, right? And they should understand why. And then that higher level broad question, right? How can we ensure clean water for wildlife and human communities in the Shenandoah Valley in perpetuity? That's a big, broad question, right? I think that's something that we all are trying to figure out throughout basically every single aspect of our society. And then I wanted to finish off this one section here before we talk a little about some of the rest of our learning community with a really fantastic visual that I just like to develop anywhere I want to, or I can, because I think it's a really fun one. This shows the cognitive hierarchy that Bloom's taxonomy on the right, and then it's a matrix with the knowledge dimension, which is going from our concrete factual information all the way up to our metacognitive information here on the left. And basically what it does is it kind of shows that throughout the learning cycle and process, our students are having to build up from the very basics, right? So if they're using factual information to learn, they can list primary and secondary colors, right? Kindergarten, very basic level. But right, by the time they're in middle school, they should be able to apply procedural knowledge to carry out our pH tests of water samples, right? So they move up pretty quickly, but they need those basic things to be able to understand and be able to build upon. Then, by the time they're in high school, they may be able to, and honestly in middle school would be great, better, to be able to determine that relevance of some of those results. So do those results actually answer the question that they sought to answer, right? And do they have the evidence to say whether or not that answer is correct? And then after that, again, we're looking at some of our higher level, metacognitive, higher cognitive level theories. So we really like to use a lot of those different teaching tools and kind of curriculum frameworks to build out our planning so that our students get to come out and learn about lots of different things and stay engaged. But we also like to make sure that all of our students who come out have an opportunity to learn. So we like to make sure that our community, we have a community of inclusive learning, which is really important for inquiry-based learning, because if students don't feel supported and safe, they're not going to take intellectual risks, and that's going to follow them throughout their career and their professional learning, or so their academic career as well. So we want to make sure that they feel like they are able to ask any questions that they may have. But we also utilize as many different accessible teaching practices, tools, and methodologies as we can. So students with any physical or mental disabilities are able to come out to Blandy and experience the learning similar to any other student. But because we have so many different students coming to Blandy from across the learning spectrum, we try to use a lot of different teaching modalities. So this is what we call the VARC learning model. This basically just means that when you're teaching, you should use a lot of different visual aids, auditory aids, reading and writing aids, and then kinesthetic aids as well. So I like this picture. This is our mammal lesson plan with our kindergarten age students. And so one of the first activities that they do about learning about mammals is they do one that involves a lot of kinesthetic and then visual. So they get a plaster of Paris mold of a mammal footprint. They get to count those paw prints or paw pads. They'll draw an example of that, and then we actually get them to go up and work with their groups to draw a graph as well to show how many mammals have different levels of paw prints. So using a lot of different teaching modalities so that they can get use some different abilities and skills. And then as well, this is them on the sniff trail. So they're sniffing a canister, and that allows them to collect some answers to then be able to put together this puzzle, and that allows them to figure out what mammal that they were originally. So we give them some puzzles and different kinds of riddles and different kind of different activities that they can actually get their hands on. They can get to use different senses, like their sniffer, as well as their sight, to be able to kind of get investigated and get engaged. And then finally, we want our students to engage with our research here. We are a field research site, and so we really strive to have our K-12 students engage with the research that happens here at Blandy, whether that's working with our undergraduate or graduate students in the summer camps, or utilizing a lot of different research opportunities here that happen here at Blandy. But we also want to showcase that science happens everywhere here at Blandy. Science is a community that is open to all, and so it doesn't have to happen someplace far away. It doesn't happen in Charlottesville or Richmond, right? We have researchers here, like Willow, like Mia, like Ethan, who are going to be out there that they can see, they can talk to, and they can understand that they can also be a scientist. And so when they come out here, they might do activities or programs mirroring some of the things that our researchers did, like a squirrel behavior study or bee pollen load activity. They might use some of the same tools like binoculars or sweep nets, water soil probes, things like that, because we want them to feel like they are, again, contributing to science. This is not just busy work where they're just going to come at the end of the day and then everything gets thrown away. Right? They're coming out here to collect some of that data, to explain it from a phenomenon, and then be able to explore some of those other questions that they have. All right, well, originally I was going to say we get outside, but it might be a little bit too warm. So we might, we can go ahead and maybe potentially shift. But before we do that, does anyone have any questions? Yeah, it's a good question. I think, from the research and things that I've seen, there could be a number of different reasons. At least in the early 2000s, there's kind of theorized that it's because there wasn't a lot of female professionals in the field. As that's shifted and as we've seen more women enter the STEM fields, we still haven't necessarily seen that interest in STEM continue to grow like we would have assumed. So it seems like now the kind of leading assumption basically is that it's kind of a mix of the teaching modalities, kind of pushing down girls and making sure that they feel like they don't necessarily have the opportunity to go out there and investigate, so their questions aren't being heard, they're being called on less in classes and things like that, so it kind of basically just squashes their voice. And then historically also, not necessarily having the same opportunities, so it's the same internships or level 2 internships or fellowships or some of the entry-level positions, and so not being able to have those like high school or college level entry-level positions into the STEM pipeline also was kind of something that they saw as a potential problem. So I think there's still a lot of research out there to show what needs to happen, but it does seem like kind of more the more that we have these inquiry-based learning and the more that we allow students to have more of a voice, that we're seeing more women and girls stay in STEM.
Are there any parts of the Arboretum's collections or grounds that you would like to see incorporated more in your instruction, either because of your personal interest or because groups are asking for it?
That's a great question. Yeah, we utilize a lot of different spots here on grounds. We primarily go up to the Peatwood area, PLC, which is the Park for Learning Center, and then down to Lake Georgette. I would love to be able to utilize the HL Pavilion a little bit more. Part of the issue with that is just walking students around the site can be problematic. Not problematic, it just takes a while. Walking around with a group of 30 kindergartners just takes longer than you would anticipate. So if you only have them out here for 2 hours, walking them out to the pavilion can take 20 plus minutes. And then they only have an hour to do their investigation before they have to come back. And then everything with bathrooms, again with kindergartners, takes a while. So yeah, I would love to be able to utilize the native plant trail more in general. But also part of that is getting facilities out there and then figuring out a way to have transportation there, yeah. That's a good question.
I have a question. So, I remember teaching at the college level. We would do group projects, and by that point, many of the students had experiences in their academic careers working with groups, and maybe they had some bad experiences. So they were the person in their group that ended up doing all the work, and nobody else contributed. Or maybe they did part of the project and somebody else took credit for it. And I'm wondering what kinds of comments or things you're hearing at different levels about students' attitudes, working in groups versus not. And maybe they don't get a choice at that level.
Yeah, that's a good question. So yeah, not every student wants to work in a group, and some students who want to work in a group might not best work in a group. And so sometimes, yeah, some of those conversations can be best to have had with teachers beforehand, just so we have a kind of inkling as to who might be somebody who we have to keep an eye on. But yeah, I think realistically, a lot of it means just mixing up different group rotations. So we try to make sure that while we might have a small group of 3 or 4 going through and working on part of the investigation, doing the actual experiment. We might have them do some of the data analysis by themselves or in pairs and break them up and reshape them. We might have it all come back together to do some of this bigger group work. So trying to make sure that it's not just all in one thing, so everyone stays kind of in those little cliques and stuff like that. But also giving students to have the opportunity to have some independent time. Because I know everyone gets tired sometimes of talking to people. So yeah, so it's really just mixing it up as much as possible. And that way we kind of allow students to have also their own opportunity to talk to some of their peers who they might not otherwise get a chance to.
Do the activities that are presented to these young people Does it require preparation on the part of the teachers and the students before they come here, or is that grade level dependent?
That's a good question. Yeah, so not necessarily. Oftentimes we want to have students be able to come out here and just do whatever work they want to do, and not necessarily have to kind of preload some of that work with them. If they have the opportunity to do that, it's usually much more beneficial, right? It allows them to get more out of it, stay more engaged, and be able to learn and take some of that more back if they're able to, you know, investigate some of those things beforehand. But otherwise, they are not required to by any means. But we do have a number, all of our programs have like 2 to 3 activities that they can do both pre and post as well.
Okay. And the teachers are aware of it?
Yes.
They know what's going to happen before they get here?
Yes, yep, absolutely. We work with all those teachers, Emily especially, who's our fantastic lead environmental educator. She works with all the teachers to make sure that they have everything that they need beforehand, so that they are all set up and ready to go. And then you know, we communicate with them multiple times, usually sometimes months beforehand, so that they know. But yeah, it's a lot of work to make sure that the students and chaperones are also aware of what that's happening.